The motivation for us in producing NetPyNE was to be able to handle multiscale modeling. And initially this was the electrophysiology, and now more and more the topic that we just heard about from Robert, which is the reaction diffusion, intercellular and extracellular. Again, particularly important for clinical applications where you deal with drugs which are given, which diffuse, which then make a change at a receptor, which often then have an entire cascade of metabotropic effects, altering calcium levels, cyclic AMP, G proteins that are really difficult to intuit what they're going to do. We have worked with this simulator, and Salvador, again, is the person who developed the simulator, well, the wrapper around the neuron simulator. Neuron was, of course, developed primarily by Michael Hines, first to manage a simulation of area M1 of neocortex. So we'll see that as an example as we go through. The neuron simulator is the base for what NetPyNE does. The reasons for using it were because that's what we were already using, frankly, but it's also is widely used, and it does it already encompassed a number of scales, and again, with this addition of reaction diffusion, we've now extended to considerably more scales, as well as taking on our own variant of LFPI, which is Gauss-Heineville's program, to look at LFPs and also broader field potentials. So this is similar to what was just shown by Robert, that there's a variety of simulators out there that ideally you'd like to test a simulation if you're going to identify it as a credible simulation in a variety of simulators and not just in one simulator. The idea is to get massive simulations because the brain is a massive device. The cortex is a massive device. 1 cubic millimeter of cortex is a massive device. So it's difficult to manage all of the parameters that go into that. It's difficult to manage all of the output that comes out of these simulations. And we work closely with the Human Brain Project, with the Blue Brain Project, with the Allen Brain Institute to try to help them and to have them help us, frankly, because they have a lot of resources, both of those, all three of those, to develop these large simulations. And more we'd like to be able to go back and forth between their resources and our resources and do that. So right now, I'd say the Human Brain Project or Blue Brain Project can do really the largest simulations that are currently available. But more and more, as high-performance computing becomes available to everyone, it should be possible for even people in smaller institutions to be able to do these. The key point about a declarative language as opposed to a procedural language is that you can simply read it and not have the procedural calls getting in your way. So here on the right, we see this for GID loop, which tells you that, okay, we're going to create cells, we're going to take random locations for their Y locations, we're going to give them a name, and we're going to then create them, basically. So here, all that's going to be done for you with a relatively simple declarative language, a declaration. Population parameters for the excitatory cells of layer 2 are going to be of type pyramidal cell. They're going to be randomly assorted between locations 100 and 400, the Y location being depth location from the pia to the white matter, and the number of cells we're going to create is going to be 50 in this case. So you can read it. It's relatively easy to export. It's relatively easy to import from, for example, SONATA or NeuroML, and it should be easily reproduced across different systems. So the other key thing is to be able to parallelize. So we've made it so that one can go directly from the serial implementation to the HPC implementation with effectively the toggle of a switch, and also so that it's relatively easy to do batch parameter explorations, which otherwise can be a lot of coding for very little, satisfaction and happiness. So that having this all handled for you on the back end is really very useful, especially if you're not of a CS background. Even if you are, it just takes a long time to write all this stuff. So again, this makes things a lot easier. So that's the package that we use, and I'll hand this over now to Salvador, and I'll give you the screen to continue. Stop share.
Okay, thanks. Okay. Can you see my screen now?
Yes.
Yeah. Okay. And go back here, and I'm gonna try to put this in present mode, see if it doesn't mess it up. Is it still working?
Yes.
Okay, great, thanks. Okay, so I'm going to give an introduction of NetPyNE and some of its features. So as Bill mentioned, this is a package to facilitate development, parallel simulation, optimization of parameters, and some analysis of networks that are built in Neuron. So before I go ahead, I just wanted to ask all the participants to actually load the GUI that we're going to use, because it might take a few minutes and we might have some issues because there's a lot of people here. So if you want to go ahead and just load this link here, it's www.netpyne.org/GUI, and you should get something like this screen. You don't need to do anything right now, just make sure that it works and it's ready for the next couple hours. So I've also uploaded the slides that we're using that contain all these links and all the useful information and step-by-step instructions of the tutorial that we're going to do. So that's available at bit.ly/cns20-netpyne. So you can also download these slides. Both of these links, the actual GUI and the slides, are on the chat. So yeah, if you have any issues, if you don't remember the link, just ask in the chat and someone will share them. So as Bill was mentioning, NetPyNE is built on top of Neuron. So here's an example of Neuron and some code in Neuron to create a synaptic connection. So we see that we have to create this synaptic object, the exponential synapse. Then we need to create a netcon and set the parameters. And so this is an example of how you would do it in NetPyNE, just to compare this sort of procedural way to the declarative. So in the declarative way, we are creating what we call a connectivity rule. So in this case, we can create thousands or potentially millions of connections at the same time, just by specifying a rule of connectivity. So here we are saying we want to connect the sensory population to the motor population with a specific probability of connection and a given weight. We can also define mathematical expressions for the connectivity. And so as you can see, this is a high-level way of defining many connections at the same time instead of having to define each one individually. And the next step that we build on top of NetPyNE is the NetPyNE graphical user interface, which is what we're going to be using in the first part of the tutorial. And this is just a way of defining these same connectivity rules, but using a graphical interface. So everything that you can do programmatically, you can also do in the interface. Here's a summary diagram of NetPyNE. I'm not going to go into much detail, but just to show the different sort of components that we have. And the first main one is here on the left, that we call high-level specifications. And that's the main one where the user is going to be involved. And it involves defining all the parameters of the network that you're going to build. So all these, the properties of the cells that you're going to use, the connectivity rules, the stimulation that you want to provide to the network. Here it's useful that we can actually import existing neural models, so you don't have to encode them in the NetPyNE language by yourself. You can run a function that imports existing HOC or Python cell models, also from NeuroML. And the next steps are things that mostly that NetPyNE automates for you. So based on these high-level specifications, NetPyNE will create a network, a neuron network for you with all the neuron objects required to run the simulation. And it will also allow you to run the simulation parallelized already with a simple command. NetPyNE will take care of all the parallelization steps that are required internally and return the simulation results from the neuron simulator. And the final step you can do with NetPyNE is this step of analysis and saving, where you can plot many commonly used functions from connectivity matrices to raster plot, LFPs, information flow measures. They're all built into NetPyNE, so you can just call a function and obtain that figures. And you can also save to different formats and export to these standardized formats in the field, such as NeuroML and SONATA. So this is just the different components that can be defined in NetPyNE. We're going to see this in the GUI later on, so I'm just going to step over this. Just to mention that NetPyNE has RxD integrated inside, so you can define RxD components for different cells using the NetPyNE declarative language, and those RxD components will get instantiated when you create the network in different cells. So this allows you to define networks at all these different scales, from the molecular level to the cell to the network, using this same declarative language. One of the strengths of NetPyNE is its flexibility to define connectivity. So you can define rules based on properties of the pre- and postsynaptic cells, such as the location of the cell, different cell types, and you have a number of predefined functions that you can use, such as probabilistic connectivity, convergence, or divergence. We also include learning mechanisms, such as STDP and reinforcement learning, as well as gap junctions. As I mentioned before, NetPyNE takes care of the parallel simulation of the network, which usually involves when you have a supercomputer or a multi-core computer, you have to divide, sort of tell which nodes in your supercomputer are going to simulate each of the cells. And once the simulation finishes, you have to gather the information back from those nodes. So that is taken care by NetPyNE. This is just an example of the different analysis functions that you can use, different connectivity matrices and ways to represent connectivity, raster plots, traces of different variables such as the voltage or currents, and as well as statistics, spiking statistics. We've also incorporated the use of local field potentials. So these are calculated by summing all the currents of all the segments in the neuron. So this is a common method also used by other tools such as eLife or the Allen Brain tools. And here's an example of calculating this LFP signal for a single cell as well as a network. So you see that you get the sort of raw LFP signal, and you can then calculate other things like the power spectral density or a spectrogram. One key feature of NetPyNE is that it allows for what we call batch parallel simulations, where you want to run not a single simulation, but many of them at the same time. This is usually done either to explore a number of parameters. So you want to test what is the effect of having different connectivity weights on your network. So you can tell NetPyNE a list of values, usually called parameter sweep. You can tell what variables you want to explore and what values you want to test, and NetPyNE will run all the simulations automatically for you and return the results. And for this, you can use predefined setups that you can customize to submit jobs either on your local machine or on different supercomputers. You can, of course, then analyze the results. And here's an example of a 4-dimensional parameter sweep where we were exploring 4 different parameters. And then you can visualize the effect of these parameters for example, on the firing rate of a specific population in the network. The other thing that's a key element of NetPyNE that we've been developing for several years now is this graphical interface. And this has been shown to be very useful for students and beginners, people who are learning to use the tool, because it gives you an idea of the different components and properties that you can select within NetPyNE. And it gives you a good introduction to then building your own models programmatically. So it's also useful for prototyping new models. If you want to start building a network, and you can build like a small scale version of it in the GUI, and then you can actually export the code that's generated by the GUI, and you can continue your model programmatically to make it like larger or do more complicated things. And it is also useful to explore existing models. So in theory, you can load any existing NetPyNE model in the GUI and just explore the connectivity or the locations of the cells, plot different analysis functions, just by loading it into the GUI. So I wanted to mention some latest updates in terms of the GUI. We have now a grant, like a voucher from the Human Brain Project to integrate the NetPyNE GUI and NetPyNE itself in their new eBrains platform. This is what the Human Brain Simulation platform is now going to be called. So it's going to be integrated into eBrains. So everybody will be able to go to this platform and run NetPyNE models there. And we hope to fully integrate the GUI as well more tightly. Additionally, something that's already quite advanced and will be available very soon is this integration with the OpenSourceBrain. So the OpenSourceBrain is this platform that provides sort of database of different models, as well as connection to GitHub, the repos of these different models. And they're now building their platform to be able to simulate, to create and simulate and analyze models online. So the tool that will be used for that is NetPyNE, but it will be integrated with the backend file management system of OpenSourceBrain, so that you can create your own workspaces where you can have multiple files, save results of your simulations, and go back anytime that you want. So you have your own personal workspace to work online with any of the models available in NetPyNE. So this is an example of the prototype where you see on the left-hand side, you see your different workspaces with different files, and the right-hand side is the NetPyNE GUI that we're going to be using today. Additionally, OpenSourceBrain is also building a tool called NWB Explorer. So this is a new format called Neurodata Without Borders. And so the idea is that it will become the standard to store and analyze neurophysiological data, such as voltage traces shown here. And the interesting thing is that because both NetPyNE and NWB Explorer will be hosted on OpenSourceBrain, it will be possible to output the simulation data from NetPyNE and also visualize it in this tool, NWB Explorer, as well as compare directly to experimental data. So you will be able to compare your simulation results to experimental data. I wanted to highlight here that much of this work, including OpenSourceBrain, the NetPyNE GUI, and the NWB Explorer, is being developed in collaboration with Metacell, which is a great company we've been working on for many years now. And you can check their website with a demonstration of many of these tools online, metacell.us. One other feature of NetPyNE is that it's compatible with these two standards that have emerged in the field, SONATA and NeuroML. Both are standards for sharing both single cell and network models. And this allows models built in NetPyNE to be imported into SONATA or exported to Sonata and then imported by other tools, such as PyNN, for example. And that should give you the possibility of running models in different simulators, such as Nest or Brian. So here's an example of a model specified in Sonata format that was visualized using tools here in the left from the Bluebrain project, as well as the NetPyNE GUI here on the right, using the same source Sonata files. Similarly, here you see the simulation output of a Sonata model using the tool from Brain, the BMTK, as well as from NetPyNE. So you see that the simulation output is identical. And similarly, for simpler models, Sonata was used to test reproducibility on BMTK, PyNE, and NeuroML. So we have a website, netpyne.org, that contains a lot of documentation, and that's been recently updated. So it's quite thorough now for all the functions and specifications available in NetPyNE. It also includes a number of step-by-step tutorials. Right now, these are mostly programmatic tutorials. So you have 9 of them to do different features that you can do with NetPyNE. And they build on complexity. So starting from 1 up to 9, you can learn from building very simple networks to more complicated networks, and then plotting LFP, running batch simulations, etc. So it's all on netpyne.org. We also have 2 Q&A forums with quite a few questions and answers already. So if you have any issues, you can check there and post any questions. And I think I didn't include in the slides, but we also have a mailing list, which is just netpyne.org/mailing. We did publish this tool, the NetPyNE tool, last year on eLife. So that paper is available online for free, and it gives you more details on this tool if you're interested. And I wanted to briefly mention now the number of models built in NetPyNE has been growing very rapidly in the community. So over the last couple of years, we have seen and worked together with many different institutions and labs who've been using NetPyNE to build models of many different parts of the brain. Here's a bunch of examples: thalamocortical networks, somatosensory cortex, visual cortex, cerebellum, modeling some specific phenomena such as ischemia or TDCS stimulation. Also some disease such as epilepsy. So the list of models, the full list of models, is available at netpyne.org/models. And many of these contain links to the different repos where these models are hosted. So you're always welcome to find a model that interests you and use that as a starting point for your models. And finally, just wanted to show a couple of examples of models that we've been building using NetPyNE and are very close to being published, several of them. So this is the one that I've been working on personally for the last couple of years. It's a model of motor cortex microcircuits in the mouse. It's a volume, a cylindrical volume of about 300 micrometers by 1.3 millimeters. You can see it here represented. Contains 10,000 neurons of 5 different cell types, 15 populations, and about 30 million synapses. So we included the real densities of cells within this cortical volume. And we've had pretty good success replicating experimental results from different labs, in particular from mouse in vivo results, showing the dynamics of mice, activity in mice when they're moving versus when they're quiet. So we also reproduced oscillations in vivo and several things. So there's a bioarchive link to this paper. I forgot to put it here, but I'll put it in the chat later. Another big collaboration we've done with NetPyNE is with the Human Neocortical Neurosolver, which is a grant from Brown University led by Stephanie Jones. And the aim of this project is to essentially explain the source of EEG signals and MEG signals based on biophysical models of brain circuits. So to do that, they simulate the current dipoles generated by physical circuits in cortex and generate these EEG-like signals. So this model was implemented in Neuron, and we converted the model to NetPyNE in order to make it more scalable, easily extendable, and to customize some parameters, as well as to build new models of different regions. So this is now available online as well. And another key example for us is this Potjans and Diesmann Thalamocortical model, which is very popular in the literature, and it's been implemented in both Nest and Brian, and we just submitted an implementation in NetPyNE, which would allow also to use, instead of point neurons, which is the original cell type of the model, we should be able to use more morphologically detailed neuron models for this network. So it includes 80,000 neurons and about 300 million synapses. The NetPyNE implementation is available in GitHub, and we've run successfully these simulations on Google Cloud supercomputers. And replicated, we have a full paper on this, just showing that we replicated the original Nest simulation results. So finally, just as a summary slide, the idea of this multiscale modeling is to essentially integrate experimental data, all the experimental data that's been generated over the last years, in order to study multiscale dynamics in these circuits, information flow, the underlying biophysical mechanisms, and eventually develop hypotheses that can be guiding for experimentation. So we work with a couple of collaborators where they have provided us data to build the models, and then we provide back some hypotheses of things that they can try experimentally. And so that sort of closes the experimental computational loop. And of course, eventually we would like to use the models to understand disease and evaluate different treatments potentially, both pharmacologically or neurostimulation treatments. Thanks, of course, to everyone that's helped building this tool, and to all our funding, NIH, New York State, and NSF.